My name is Juliana Acosta-Uribe. I am a researcher at the University of California, Santa Barbara, and I'm also part of the neuroscience group of Antioquia. We are researchers in neurodegeneration in the Universidad de Antioquia, which is in Medellin, Colombia. I come from the frontotemporal dementia field. I've been researching Alzheimer's disease and FTD for more than 10 years, and we had this really incredible project where we were chasing families with genetic dementia throughout the country, through Colombia. And then we found an amazing set of individuals that had both FTD and ALS within the same family. Turns out that they carried a very unique mutation in a gene called TARDBP, which encodes for TDP-43, which is a key protein for almost all sorts of neurodegeneration. You see a role of TDP-43 in ALS, frontotemporal dementia, even in Alzheimer's disease, there's a role for this protein. TDP-43 is a ubiquitous protein in different neurodegenerative illnesses. We've said that ALS and FTD is just part of the same spectrum. You can have one without the other, or you can have both. They share a lot in their genetic architecture, the proteins and genes that are involved as in, let's say, intermediate steps, and the routes that lead to neuron death. TARDBP is a gene that it's fascinating because if you look in the literature, it's usually associated mainly with ALS. But that is because most of the literature that we read comes from scientists that are based in the United States, Europe. But when you look in a broader spectrum throughout the world, you're going to see that TARDBP, it's associated with FTD in Asia, in Africa, and in Latin America. In a recent study that we clumped different sets of data, we found that it was the most common gene associated with genetic FTD. C9orf was only a small part. TARDBP was everywhere, and we even saw the same mutation arising in 2 different countries. I studied some families that have a variant called I383V, and we saw the same variant popping up in families in Colombia, in different regions, and also in the south of Brazil. We were thinking, this is incredible. We know it's a recurrent mutation in the gene, but we didn't expect it to be so widely spread throughout Latin America. The center that I come from has been following a family with Alzheimer's disease for over 30 years. The same kindred, the same mutation, and we have been able to learn so much about the transition from a healthy brain to a brain with a neurodegenerative illnesses throughout all the different stages. We've been able to follow individuals since they were children all the way until they have advanced dementia. When we came across these unique families that have the FTD ALS spectrum, we said, why not? Let's leverage on all that we've learned with longitudinal studies of single families and move it to a different gene. It's everything that we had learned in terms of long-term follow-up of individuals. We said, let's do that with individuals that carry TARDBP mutations. Let's try to stay with them the longest. Let's try to just be a part in their lives, check up frequently, and also let's partner with ALS to figure out how can you determine when someone is about to start the process of changing from a healthy individual to either motor symptoms or cognitive symptoms. We call that phenoconversion. What is the key point where we can know that someone that is a carrier of a mutation will start having symptoms? That is the perfect moment to treat, ideally, because no neurons have died massively, and we still have a lot of either cognition or movement to preserve. You don't want to treat someone since they're like 10, waiting to see if they're going to develop the illness at 30, 40, 80. No, you want a moment that is crucial in the process that you say, if I intervene now, I can either stop the process or at least stall it for as long as possible. We also have very interesting questions to ask these families. One of them is, what determines if an individual would follow the ALS route, the FTD route, maybe the FTD ALS, and singularly, what determines that a carrier will live a healthy life until their late 80s. We've had a couple of outliers, and these are coincidentally all women that were able to live absolutely healthy till their mid to late 80s, which is like, okay, they know something, or they have something we ought to know. Because by studying these outliers, we can clearly identify something that could act as a key to either stopping or stalling the neurodegenerative process. Both of the approaches of studying different people from different ancestors, different backgrounds, and follow them up versus studying a single family, both of them have pros and cons. However, studying a single family allows you to reduce the noise outside. You will have people with a very similar genetic makeup that grew up in the similar conditions. You have shared risk factors, both of neurodevelopment, exposure, habits, food you eat. So you're reducing a lot of variables. You're making them less variable, steadier in individuals. It also allows to know who has the greater chance of getting sick. If you're studying the global population, you don't know who will develop ALS. There's still no tool that can accurately predict who will have ALS unless you know they're carriers of a genetic variant that is associated with the illness. That's when you know this person is a higher risk. And if you follow them through their life, then you could pinpoint crucial time points that then you can extrapolate to individuals that do not have genetic mutations and say, okay, if we've learned this biomarker, this metric, this test can predict when someone who is a carrier of a C9orf72 or SOD1 mutation or a TARDBP, if they have this in common before starting the symptoms, can we move that to the general population? I was approached by Target ALS a couple of years ago, and they had learned a little bit of my research in FTD ALS spectrum with these families, and they thought it was remarkable. And coincidentally, it overlapped with opening one of the natural study sites in Bogota under the work of Dr. Marta Peña. So both things happening in Colombia, I believe, led the organization to reach out to me as a geneticist and said, this could be a great partnering between Target ALS, Colombian scientists, to advance the inclusion of diverse populations in the natural evolution and the Target ALS ecosystem. I've had an incredible impression and an incredible experience of working with Target ALS team and affiliated scientists. It is really nice to see how everyone is very happy to share data, share methods, collaborate, help you in developing tools or strategies you haven't used before. Everyone has a very team mindset to solve the big puzzle of ALS. I think the ALS community benefits having some of the FTD cognition scientists kind of stepping in and saying, okay, we gotta tackle this spectrum together. It cannot be just about the cognition, just about the motor neurons. It's about everything that happens in an individual that is, you've been diagnosed with FTD. Well, you're gonna have a 40% chance to develop motor neuron involvement. You have been diagnosed with ALS. Well, you're gonna have a huge chance of cognition impairment. So we need to work together because both elements are so essential for life. My name is Juliana Acosta-Uribe. I am a scientist from Medellin, Colombia, the Universidad de Antioquia, and I am building a world where everyone lives.